name is Sophie, and I'm a rising fourth year at the University of Virginia. This summer, I'm interning with Dr. Hoadley at the Dauphin Island Sea Lab. My work is investigating the change in frequency of sexual recombination under nutrient-limited conditions in cultures of endosymbiotic algae originally isolated from coral hosts. These algae, shown as the brown spots on this coral polyp, photosynthesize for the coral host, and in exchange receive shelter and a nitrogen source. The dysfunction of this symbiosis is the cause of coral bleaching. For the first 2 weeks of my internship, I traveled to a coral farm in Ohio and the Mote Marine Laboratory's coral nursery in the Florida Keys to collect samples of coral and their symbionts. Since these corals were kept in the same water and under constant light conditions, it was a perfect opportunity to understand how host and environmental characteristics influenced which species of symbiont we found in different coral species. The photopigments used by these symbionts contribute to the diversity of colors you see here in their tanks at the farm. We collected measurements of their photosynthetic ability within host using a special fluorometer built by Dr. Hoadley, as well as preserved tissue and algal samples to analyze back at the lab. My current work at the Dauphin Island Sea Lab entails maintaining and sampling pure cultures of 8 different strains of these algal symbionts originally isolated from coral hosts. This entails collecting daily samples of the 4 replicates for each culture to track changes in cell populations and to later be used with a flow cytometer to count sexually reproducing cells. This also entails filtering and preserving samples of these symbionts to be analyzed for changes in gene expression, specifically investigating genes linked to meiosis using qPCR. And finally, taking photophysiology measurements using a fluorometer. These measurements involve placing a sample of the culture in a cuvette and exposing them to multiple wavelengths of light in order to track changes in their photosystem efficiency and photopigment orientations, which are important characteristics of their photosynthetic ability. This research project will contribute important insights about how the algal symbiont changes primary production and reproductive strategies under nutrient-limited conditions, such as those found within the coral host. Since we are analyzing multiple species of symbiont, we'll be able to compare primary production and rates of sexual recombination across the different species. Sexual recombination increases the genetic diversity of offspring, so this may be a potential mechanism of adaptation for the coral symbiont. Since these symbionts play such a key role in coral health and skeleton synthesis, they may provide an avenue of adaptation for the coral host under projections of global climate change and ocean acidification.